Hello everyone, this side Bunch Langneja here, and I'm excited to talk about an important topic, the cell cycle and the cell signaling, two processes that are essential for life. We will explore how these two work together to keep our cells healthy and what happens when they go wrong, leading to diseases like cancer. We will also touch on some of the amazing therapies that target these systems and how they are improving our lives. So let's begin it. Basically, let's start by the thinking of our body as a big organized city. In this city, every cell is like a worker, following a strict schedule and communicating with others to keep everything running smoothly. The cell cycle is like their work schedule. It tells cells when to grow, divide, or rest. And the cell signaling is like the messaging the system, ensuring cells get the right instruction at the right time. But what happens if workers don't follow their schedule or get bad instructions? In our bodies, this chaos can lead to diseases like a cancer. Cells starts growing out of control, ignoring signals to stop or repair themselves. So today we will look at how the cell cycle and cell signaling work, how they are connected, and how the system can break down and cause the disease. We will also see how scientists are fighting back with the target therapies. So let's begin with the cell cycle. So what is the cell cycle actually? We can say it is the process that ensures cells grow, replicate their DNA, and divide properly. Basically, it is the process that our cells follow to grow and divide. It's divided into mainly four phases, and each phase has a specific job. Let's see what they do. The first one is the G1 phase, where the cell grows and prepares for DNA replication, like gathering supplies for the next step. The next is the S phase, where the cell copies its DNA. Imagine duplicating an instruction manual. So G2 phase, the third one, where the cells double checks everything to make sure there are no errors like proofreading. Yep. And the last one is a mitosis, where finally the cell divides into two identical daughter cells, which we can also call it the M phase. Now, to keep everything on track, the cell cycle has the checkpoints. Let's go on the checkpoints. First one is a G1S checkpoint, where they ask, is the cell ready to copy its DNA? The next one is G2M, where they ensure the DNA is accurate before division. And the last one is a spindle checkpoint, which makes sure that the chromosomes are lined up properly during the division. These checkpoints are critical for preventing mistakes. When they are failed, damaged cells can keep dividing, which can lead to the disease like the cancer. Next one is the main cell signaling. Now let's switch gears to the cell signaling, which is how cells communicate. Imagine cells sending text message to each other. Here's how it works. Signaling molecules like a growth factors act as the message. It is sent by the one cell and received by the another one. The receiving cell has a receptor like a mailbox that catches the message, and this triggers a chain reaction inside the cell called signal transduction pathway, which carries the message to the nucleus. The nucleus then acts on the message, telling the cell to grow, divide, or stop. For example, we have two examples of it. The MAPK/ERK pathway tells cells to grow and divide, and another one, PI3K/AKT pathway, helps cells survive and prevent them from dying unnecessarily. When these signals are misinterpreted or sent incorrectly, cell might start dividing when they shouldn't, leading to uncontrolled growth. It can't be. Now, the interaction between cell cycle and cell signaling, the main concept. Here's where it all comes together. The cell cycle doesn't operate alone, it's controlled by signals from the environment. For example, when a growth factor binds to a receptor, it activates pathway like MAPK or PI3K. The signals push the cell past the G1 checkpoint, allowing it to start DNA replication in the S phase. If the cell detects something wrong, like DNA damage, the P53 pathway steps in. It pauses the cell cycle to repair the damage, or if the damage is too severe, triggers cell death. But when the system fails, it leads to a big problem. For instance, mutation in the RS gene can cause constant growth signals, pushing cells through the cycle even when they shouldn't divide. This kind of unchecked growth is one of the first steps toward cancer. Moving forward to the districulation and disease. So what happened when the system go off the rails? In the cell cycle, overactive proteins like cyclin and CDKs force cells through checkpoints without proper checks. At the same time, mutation in tumor suppressor like P53 or RB remove the brakes, allowing damage to cells to keep dividing. In cell signaling, mutation is oncogenes like RAS create constant growth signals, while the loss of tumor suppressors like PTE removes the stop signals. What is the result of it? Cells grow uncontrollably, which leads to hallmark of cancer like uncontrollable proliferation, where cells keep dividing, genomic instability, where DNA errors accumulate, resident to apoptosis, where damaged cells don't die when they should. So this combination creates an environment for a cancer to develop and spread. Let's go on the cancer. So what is it? Let's look at this some real world example of how this plays out in cancer. In HER2 positive breast cancer, the HER2 receptor is overexpressed. This means too many growth signals are sent through pathways like MAPK and PI3K, causing rapid and uncontrolled cell division. Another example is P53 mutation, which are found in over 50% of cancers. Without functional P53, damaged cells don't repair themselves or die. They just keep dividing, accumulating more arrays, and fueling tumor growth. This disruption allows cancer cells to grow unchecked and even spread to other parts of the body. Now, therapeutic implication. So, how do we fight back? Now, the good news is that understanding this process has led to breakthrough in cancer treatment. Targeted therapies block specific pathways. For example, transhuman target HER2 receptor in breast cancer, cutting off the growth signals. CDK inhibitors like palbociclib stop cancer cells from moving through the cell cycle by targeting overacting cyclins. Last third one is immunotherapy helps the immune system to recognize and attack cancer cells. Looking to the future, personalized medicine aims to customize treatment based on the patient's specific genetic and signaling profile, which could make therapies even more effective. Mm, To wrap these things, The cell cycle and the cell signaling are essential processes that keep our cells functioning properly. When the system are disrupted, they create the condition for disease like a cancer to develop. But with the advancements in our understanding, we have been able to create targeted therapies that do not only improve outcomes but also offer hope to millions of patients. Thank you so much for listening to me. I think. Thank you.